All right, everybody, so here we are for the second lecture of the course, Molecular Genetics Part 1. And before we start, just not going to lie, this is going to be a long one. Long video. You probably saw from the runtime already that this is by far our longest video of the course and a lot to get through. So as we start off here, I just want to make it known that there's no expectation whatsoever that you watch this whole video in one sitting. If you've got the attention span and the wherewithal, then all the power to you. But please feel free to break this video watching up into multiple parts and take whatever breaks that you need. Good stopping points for the breaks are the embedded questions. As the embedded questions come, once you answer them and save your response, a nice opportunity to step away and come back when you're mentally prepared to continue the video. So this is the first of 2 lectures on molecular genetics. What we're going to talk about today is overall chromosome structure, includes the physical nature of DNA as a physical molecule, the organization of chromosomes, chromatin or packaging of DNA, and the individual building blocks of that chromatin packaging, which is the nucleosome. And then we'll move on to gene expression. We'll talk about the central dogma in general and get into detail around transcription, mRNA processing, and translation, all in the effort to build a protein. Really, the only point of genes is to encode the instructions for building proteins, and the central dogma is the process by which the information contained in the genes manifests into proteins. But first, let's talk about the basic physicality of DNA as a molecule. And DNA is made up of individual building blocks called nucleotides. Nucleotides, the individual subunits of DNA, are made up of 3 main parts. Each one contains a ribose sugar, and that we see here in pink. A phosphate group, here we see that in yellow. And the actual base itself. That's going to be adenine, as we see in orange, but it could also be thymine, cytosine, or guanine, the 4 different bases of DNA. And here are those individual bases, color-coded. Again, we have adenine, this time in yellow, guanine in orange, thymine in purple, and cytosine in pink. And you can see that we've got 2 categories or classes of bases. We've got purines, which are double-membered rings, and we've got pyrimidines, which are single-membered rings. Thymine and cytosine are the pyrimidines, and guanine and adenine are the purines. And I'm sure you know already that adenine base pairs with thymine using 2 hydrogen bonds, and guanine base pairs with cytosine using 3 hydrogen bonds. We'll come back to that in just a bit. So the sugar and the phosphates, the non-base components of the nucleotide, are completely structural. They form the backbone of DNA. This is the banisters of the spiral staircase. If we think of the double helix of DNA as a spiral staircase, the phosphate sugar backbone makes up the banisters. And here we see it, phosphate, sugar, phosphate, sugar, phosphate, sugar. No information, just purely structural. It holds DNA together. That backbone, again, is repeating. It's just phosphate and sugars in a row. Those phosphates have negative groups on them, so that's what makes DNA negatively charged, and no information. The information of DNA is encoded by the bases themselves. The adenine, guanine, cytosine, and thymine contains the information for building individual proteins, and those proteins then allow life to be possible and govern all the phenotypes that we see DNA controls. So if the phosphate sugar backbone of DNA are the banisters of the spiral staircase, then the bases are the steps of that staircase. It's what holds the 2 strands of DNA together in the middle, again through hydrogen bonds. So here's a more accurate representation because it shows a bit of that spiral, that 3-dimensional twist of DNA. We've got the phosphate sugar backbone of the DNA here in yellow. We've got a sugar, phosphate, sugar, phosphate, sugar. We've got another one on the other strand here, anti-parallel. Phosphate, sugar, phosphate, sugar, phosphate, sugar. And the bases are in the middle of the DNA molecule. We've got a cytosine base pair to a guanine with 3 hydrogen bonds. We've got a thymine base pair to an adenine with 2 hydrogen bonds. The bases holding the information in the center of the double helix. That implies that DNA is double-stranded. We can bring these 2 strands of DNA together to form a double-stranded molecule of DNA, deoxyribonucleic acid. Those single strands we've already discussed. Again, the phosphate sugar backbone is here making the gray helix, the backbone, the structural component of the DNA, and we have the bases pointing inward. This is 1 strand here. This would be the adenine and thymine, guanine, and cytosine, which contain the information. These chains or strands of nucleotides, they're linking together to each other. Those phosphates and sugars are being held together in a 5 to 3' direction. The very, very beginning of any single DNA strand is referred to as its 5' end, because that primary phosphate, that very, very first phosphate, is actually linked to the 5' carbon of that nucleotide. And the very end of a single DNA strand is its 3' end. And that ends in a hydroxyl group, an OH group, linked to the 3' carbon of that base's ribose sugar. And we see that here. Here is the very beginning of this DNA strand. This is the 5' end. This is the 5' phosphate linked to the 5' carbon of the ribose sugar. And we have a thymine on that sugar. So this sequence of this strand would be thymine, adenine, cytosine, guanine, held together by the backbone of phosphate sugar, phosphate sugar, phosphate sugar. And the very end of this strand culminates in a hydroxyl group. And that hydroxyl group represents the 3' end of this strand. It is the end of the strand. And it's because it is linked to the 3' carbon of that ribose sugar. All of the bases point away from the backbone, again inwards towards the center of the helix, and those bases can pair up through hydrogen bonds, giving us our adenine, thymine, guanine, cytosine backbone. And I feel like I'm beating a dead horse at this time, but adenine and thymine are held together by only 2 hydrogen bonds, and guanine and cytosine are held together by 3 hydrogen bonds. Now, the organization of DNA goes well beyond this kind of simple sequence idea of bases on antiparallel strands. We actually have genes, regions of DNA sequence that are organized to encode the information for building proteins arranged on DNA molecules in fairly specific ways. Also, DNA in cells such as ours, in eukaryotic cells, has to be compacted in order to fit in the nucleus. We'll get back to this in just a bit, but we actually have 6 feet of DNA in every single cell in your body, and you've got about a trillion cells, even 1 trillion cells in your body. And so that's 6 times a trillion. You got 6 trillion feet of DNA in you right now, and that requires a great deal of compaction. This compaction of DNA has to be organized, regulated, monitored. It has to be reversible so DNA can be accessed. It's actually a beautiful and elegant process, this compaction of DNA. If we use any textbook as an example, we'll see that the sequence of an average human gene, and again, by sequence we just mean the bases composed of that gene, the sequence of adenines, thymines, cytosines, and guanines in that order, would take about 1 page of an average textbook. That's 1 gene taking up 1 page. The complete sequence of the human genome would require, by that same logic or that same correlation, 1,000 typical college textbooks filled with nothing but adenines, thymines, cytosines, and guanines. So what does that show us? That shows us we've got a lot of genes, we've got a lot of DNA. The human genome is vast. And again, it requires a lot of compaction to fit into a typical eukaryotic nucleus. So let's get some idea of what sizes we're talking about here. The average human cell's nucleus is between 5 and 8 microns, or micrometers in diameter. This is a 1/1,000,000th of a meter, right? This is 10 to the minus 6th meters. So very, very small. To give that some relevance, the average width of a human hair is about 65 microns. So a single nucleus is roughly 10 times narrower than a human hair. And again, the human genome is about 6 feet long. So if we were to take all of the DNA found in a single human cell, all those chromosomes, 46 of them, and line those chromosomes up end by end so that we have a single file, single chain of DNA representing all of the genomic DNA in a cell, that chain of DNA would stretch out to be about 6 feet in length. But we know that that's broken up into 46 different chromosomes. So each chromosome is far shorter than that, roughly, on average, 6 feet divided by 46 chromosomes. But still, that's a lot of DNA to fit into a very small nucleus. You can think of this as being synonymous to stuffing 24 miles of a very fine thread into the center of a hollow tennis ball. Possible? Sure, if the thread is thin enough, you can fit all of that into a tennis ball. But still, not a trivial thing, right? That just cramming all of that thread into such a small space takes some effort. And the same is true for our genome. For eukaryotic cells, we use the term chromosomes to mean very long, contiguous, so single molecules of DNA that are packaged, that are condensed in some way. And chromosomes are packaged so that they can fit inside a cell's nucleus. They're organized so that the cell knows exactly how and where to access genes. And again, it's going to be reversible so that those genes can be accessed when needed. And that means access for replication, so the genome can be copied, for gene expression, like we'll talk about near the end of this lecture, and also for DNA repair. As DNA becomes damaged and mutated, we need to access those regions to fix them. We also see some of the deepest compaction of DNA for mitosis. Mitotic chromosomes are among the most tightly compacted forms of chromosomes, and that's so that those chromosomes can be dealt out to daughter cells without breakage or tearing. I'm sure you've learned about mitosis in other classes, and we won't discuss it here. So we've got some mental idea now of the organization of DNA, right? We know that DNA is made up of long chains of nucleotides. We've got two chains or two strands of nucleotides that come together in an antiparallel way through base pairing, and hydrogen bonds are holding the two strands of DNA together to make a single DNA molecule. Along that DNA molecule are specific islands of sequence, regions of sequence that contain the information for building protein. We call those genes. And these long chromosomes must be compacted to fit in the small nucleus. So, at least from me as an instructor in past experience, I have found that students at this point begin siloing their thinking. They think of DNA as one thing, genes as something completely different and isolated, chromosomes as a third thing. And before we get too much further into the terminology, I just want to dispel you of those misconceptions, if they are in play here, and bring these together into one cohesive way of thinking. So all of these are the same thing. Chromosomes have genes in them. DNA make chromosomes. Genes are made of DNA. Like DNA, genes, and chromosomes, they are all largely synonymous. It's all DNA. So how do these terms actually fit together? Well, DNA is always going to be long chains of nucleotides strung together in a particular sequence. That's what DNA is, a double-stranded helix of nucleotides. Some of those particular sequences contain the information for building proteins, and we call those regions of DNA genes. But genes are segments of DNA. Genes are just sequences of DNA, just as all DNA is sequences of DNA. Genes are just sequences of nucleotides, just as all DNA is sequences of nucleotides. It's just that those particular sequences of nucleotides in genes hold the information for making proteins. Now, these long molecules of DNA that contain these information coding regions for making proteins, they are too long to just hang loose, and so they have to be condensed, they have to be wrapped up, they have to be compacted. And when they do that, we call those compacted regions chromosomes. So chromosomes are wrapped up DNA, and in that DNA are sequences that contain the information for building proteins or genes. So here it is visually. Here we have a cell. Within the cell, we've got that 5 to 8 micron nucleus. And in that nucleus are lots of chromosomes that are wrapped up really tightly like this one. So this is very, very tightly compacted DNA. And here's one string of that DNA that we can see and kind of zoom in on. So it's made up of two chains, two strands. The phosphate sugar backbone is here in this kind of dark purple ribbon, this dark blue ribbon. That's the phosphate sugar backbone. And we see the bases pointing inward. We've got cytosine, thymine, guanine, and adenine. And we see that they're base pairing. Adenine is base pairing with thymine, and guanine is base pairing with cytosine. So here's our long chain of nucleotides making our double-stranded DNA. And that double-stranded DNA can be wrapped up tightly into a chromosome, and that chromosome fits in a nucleus. And in this context, we see a gene. A gene is just a subset of that DNA chain, of that DNA molecule. But in this region that we see here, there is the information for building a protein. There's something in this sequence that lets the cell know that this is information for building a protein, and the cell can access this DNA and use that information to do protein synthesis. So the gene is just a particular region of DNA that we see here within the larger context of the full DNA molecule, which is being wrapped up and condensed into a chromosome so it fits in a nucleus. So again, it's all DNA, it's all one thing, and hopefully if there were any kind of misconceptions or just that idea of like, what do these terms mean? We're getting a little Traxoline here. Try to give some clarity around that. So we use these different terms just so we know what kind of DNA we're looking at. If we say chromosome, we know we're looking at compacted DNA. If we say gene, we know we're looking at information coding DNA. When we just say DNA, we mean any and all strands of nucleotides. So by using terms like chromosome and gene, we have a little bit of a finer, more nuanced definition of the DNA that we're looking at. All right. So now let's talk about some of that compaction. And the basic subunit of eukaryotic DNA compaction is the nucleosome. And personally, I love nucleosomes. I got my PhD studying chromatin compaction and how it influences gene expression. That's what I studied for my degree. I've spent almost my entire professional research career on studying how genes are expressed in the context of DNA compaction, so this stuff is near and dear to my heart. The fundamental components of this compacted building block of DNA of a nucleosome are called histone proteins. So we're going to kind of deconstruct DNA compaction and then reconstruct it once we understand what all the players are. So histone proteins are the proteins that allow DNA compaction to occur. There's 4 different types of them. And they come together to allow a nucleosome to form. There's also a 5th histone protein that we'll talk about a little bit later. But the 4 main histone proteins are called H2A, H2B, H3, and H4, where the H stands for, of course, histone. And each nucleosome contains 2 copies of each one. So we've got 2 identical copies of H2A, 2 identical copies of H2B, 2 copies of H3, 2 copies of H4. So that means we've got 8 total histone proteins that come together to make the center of a nucleosome. And we call that an octamer. It's a protein complex made up of those 8 individual proteins. And this histone octamer forms a spool, a protein spool. And maybe you now kind of can see where this is going. If the histone protein octamer forms a protein spool, then we need DNA thread to wrap around the perimeter of that spool. And that's how we compact DNA. Just like you would take a clump of sewing string, and if it was this disorganized mess in a lump, you could organize that by wrapping that thread around a spool. Now you got a nice orderly, kind of non-tangled, non-knotted, non-dangerous, non-fragile version of that thread. The cell does the same exact thing with DNA. It wraps that DNA around this protein spool. So the DNA wraps around the outer edge or the outer perimeter of this histone octamer, and very analogous to thread around a spool. And that's our first level of compaction. So here we can see it visually. We've got 2 copies of H2A, 2 copies of H2B, 2 of H3, 2 of H4, and they come together to form this histone octamer core, this histone complex made up of those 8 proteins. And we've also got about 150 base pairs of DNA. That 150 base pairs of DNA wraps neatly and cleanly around this histone octamer, and this is your nucleosome. So a nucleosome consists of your histone core and about 150 base pairs of DNA wrapped around it. And that's our first level of compaction. So that nucleosome, the 8 histone proteins and the DNA wrapped around it, is the smallest fundamental building block of DNA compaction of chromosomes. And we define that as a nucleosome again. Now, as the DNA exits one nucleosome, it leads to the next. And the DNA that links one nucleosome to another is naked DNA, meaning it has no proteins on it. And we call that linker DNA because it links nucleosomes together. This pattern of linked nucleosomes is basically what allows us to control DNA compaction. And the typical metaphor or analogy that's used in molecular biology to describe this is beads on a string. So now here we have some nucleosomes linked together, and we see the linker DNA in context. So we've got 3 nucleosomes in front of us here. Here the histone octamer core is in yellow. So we've got 1 histone core, 2nd histone core, 3rd histone core. We've got DNA coming in here, 150 base pairs of DNA wrapped around this core. And then most linker DNA averages about 50 base pairs in length. So here's another 50 base pairs. We're up to 200 base pairs of DNA total. 150 wraps around the 2nd histone core. And this brings us to 400 total DNA base pairs, right? Because we got another 50 of linker. And then another 150 wrapped around this histone core. So these are your beads, right? On the string, on the necklace of DNA. And I can't emphasize it enough. This is one what did I say? It's about 400. This is 550 base pair length of DNA. It's 150 wrapped around the first histone. 100 brings us, the 50 base pairs of linker DNA brings us to 200. Another 150 on the second. Linker DNA brings us to 400, and another 150 on the third. So this is 550 base pairs of DNA, all one DNA molecule, wrapped around these 3 histone cores, giving us 3 nucleosomes. This is a true electron micrograph of nucleosomal DNA. So this isn't a textbook schematic. This is what we can see in a very high-power electron microscope. Again, beads on a string. Each of these dots is an individual nucleosome, and amazingly, we can even see evidence of the linker DNA between those nucleosomes. I won't count them here, but we probably have somewhere on the order of 40 to 50 nucleosomes that we can see here, this beads on a string idea. So just so we don't get too bogged down in the schematic, I do want us to appreciate that we are looking at individual proteins. This histone octamer core is not a yellow disk. It really is made up of 8 individual proteins, which we can see color-coded here. And the DNA wraps around those bases, around those histone proteins, with 146 bases per nucleosome. The other thing to point out here is that we have these amino terminal regions of the histone proteins that project or point out of the nucleosome. Proteins have a beginning called the amino end and an end called the carboxy end. So the very, very beginning of a protein is its N-terminus, its amino terminus, and the very, very end of a protein is its carboxy terminus or its C-terminus. And here we see kind of the beginnings of these histone proteins projecting out like loose string from the histone proper, from the nucleosome proper. And those histone tails are going to become important a little bit later on in the lecture. Here's another beautiful space-filling diagram of a nucleosome. In fact, this is my wallpaper on my work computer at Bay Path. It's just a beautiful, beautiful image of a nucleosome. We've got the DNA, of course, in silver and green wrapped around the perimeter, and we can see the histone proteins inside the histone core there. And here's one of those amino terminal tails projecting out. So we already said that DNA is negatively charged, right? Because the phosphate groups in the DNA backbone have a negative charge, and histones are largely positively charged. That's a nice electrostatic interaction, right? The negative backbone of DNA is attracted to the positive charges of histones, so histones and DNA want to interact. Opposites attract. They interact with each other magnetically, or more accurately, electrostatically. So there's this favorable interaction of charge between DNA and histones. And again, histones have some of their ends pointing out, and some of those ends are also positively charged. Those are called the histone tails. Even when they're the beginning of the protein, we call them the tails. And they too have positive charges to kind of wrap around the DNA and keep it locked in place. So all eukaryotic DNA is packaged in this way. It's wrapped around nucleosomes in the way that we just described, and that's the first level of compaction. Very, very highly conserved process in evolution. Everything from yeast cells, baker's yeast, all the way to us, we wrap our DNA in the exact same way. And nucleosome compaction, what we just described, this beads on a string configuration, is responsible for about a 1/3 compaction. So if you've got 6 feet of DNA naked, just by wrapping that DNA around nucleosomes, you get that 6 feet down to 2 feet. Again, a 1/3 compaction. However, still, 2 feet is not going to fit in something the size of 1/10 of a human hair. So we need more compaction. We need higher and higher levels of compaction. The next level of compaction, beyond nucleosomal DNA, is called the 30 nanometer fiber. And it's important to point out that the 30 nanometer fiber, although it's the second level of compaction, is the least packaged, least dense form of DNA that we would ever find in a living cell. If we unravel DNA to anything less than the 30 nanometer fiber, it simply takes up too much room in the nucleus and the cell would die. So the 30 nanometer fiber, again, is the second level of compaction, but it's the least dense DNA can ever be in a living cell. So how do we get to the 30 nanometer fiber? If you imagine that nucleosomal DNA is these beads on a string, think of it as a pearl necklace. So here with my hands, I'm kind of just imagining I've got a pearl necklace in my hands. I'm holding a pearl necklace between my two hands, and you can do the same. Now stick out your pointer finger on one of your fingers and imagine that you're going to twist that pearl necklace around your finger. So you're going to get this long pearl necklace and you're going to have it take up much less room by twirling it around your finger. That's the 30 nanometer fiber. That's really all it comes down to. So the 30 nanometer fiber is the nucleosomal DNA that we see here. But we take this DNA and we wrap it as though it was wrapped around a pencil or a finger. We coil it so it takes up much less room. So that is what we mean by the 30 nanometer fiber. And here we see it schematically. This is nucleosomal DNA. The histone core here is in green, and the DNA itself is light blue. And we're taking that long extended beads on a string configuration and we're wrapping it or coiling it into the 30 nanometer fiber. So the 30 nanometer fiber is a coiled helix of nucleosomal DNA, and now we're getting to some real compaction. What's critical to forming the 30 nanometer fiber is that fifth histone protein that I alluded to earlier, histone 1. Without histone 1, the 30 nanometer fiber cannot be formed. So at the center, or the heart of the 30 nanometer fiber, is this histone 1 protein. And again, we see here as well, we're taking these individual nucleosomes, these beads on a string, and we are coiling them, we are bundling them into the 30 nanometer fiber. So histone 1, or H1, is often called the linker histone because it interacts with the linker DNA. It's positioned on the very edge of the nucleosome where the DNA is leaving the spool. So the DNA wraps around that spool, right? So here's the DNA wrapping around the histone core. Histone core is in yellow, the DNA is in red. We've got 150 base pairs, more or less, wrapped around the histones themselves. And now that DNA is leaving the histone. This is the linker DNA on its way to the next nucleosome. And H1 binds right at that junction where the DNA leaves the nucleosome. So we can see there's H1 right there in green, and it's binding right to the region where DNA is leaving the nucleosome. And when H1 binds in that region, it interacts with that linker DNA and it promotes it to kink. It makes it bend, and we see that here in this schematic as well. This bending of that linker DNA promotes the nucleosomes to coil into the 30 nanometer fiber. So by kinking or bending linker DNA, you cause these nucleosomes to take on this helical spiraling pattern and they bundle into the 30 nanometer fiber. That 30 nanometer fiber, you can think of that now as a rope, a bundled rope of DNA, is further looped and compacted and coiled in ways that we don't fully understand even now. We don't have every single detail ironed out on these higher levels of compaction. But ultimately, the highest level of compaction we can go is the mitotic chromosome, the form that DNA takes only during mitosis, during prophase and prometaphase. And that form of compaction is 10,000 times more compact than naked DNA. So take 6 feet, divide it by 10,000, and that's how much room mitotic chromosomes take up. And that can fit easily in a nucleus. Again, those higher levels of compaction aren't fully understood. Everywhere between the 30 nanometer fiber and the mitotic chromosome, we do have a very good sense of how that occurs, but we don't understand it fully. We can diagram it, though, as much as our understanding allows us to, and get a sense of how these levels of compaction relate to one another. Up at the top here, we have naked DNA. Again, it looks to be, in this case, about 550 base pairs of DNA. And we can wrap that up among 3 nucleosomes. We can take that and condense it down to about 30%. Remember, about 1/3 compaction, just by wrapping that DNA around histone cores and allowing it to form nucleosomes. This is that bead on a string idea. We can then coil those nucleosomes with the help of H1 because it promotes those kinks of the linker DNA. And now those nucleosomes will bundle into this ordered array or coil of nucleosomes, and that's our 30 nanometer fiber. This is the lowest, least dense compaction of DNA we would ever find in a living cell. The 30 nanometer fiber, so what we're looking at here is representing this very small region right here. So this 30 nanometer fiber can be further looped and bundled, so it takes up even less room because of looping and bundling. And those bundled loops of 30 nanometer fiber can then be coiled and stuffed, for lack of a better term, into a mitotic chromosome. And here's that mitotic chromosome, 10,000 times more dense and compact than the naked DNA we started with. So this is taking up, a few inches, and this is taking up fractions of a micron. These are false color, but real scanning electron micrographs of human chromosomes. So the pink and the beige is added by a computer, but this is a real image of two different human chromosomes in mitosis. These are mitotic chromosomes. And you can see two things, actually. One is just the beauty of this. Well, maybe three things. Now I'm geeking out, so here's a geek out warning, I guess. So three amazing things at least here. First amazing thing is that we can do this at all, right? That we can have such a beautiful image of something so small and microscopic as two single mitotic chromosomes. So the fact that we even have this image is amazing. The second thing is the beauty of the compaction, right? The organization of this, that cells following their genetic programming can cause this compaction to occur. The third thing we can note is if we zoom in really closely, and all I did here was I took this image of this picture and zoomed in, so the resolution goes way down. But what we see, these little hairs, these little wisps, right? They look like almost fuzz coming off the chromosome. This is 30 nanometer fiber DNA. This is little bits of loose 30 nanometer fiber DNA that kind of got away from the greater compaction of the mitotic chromosome. They are the threads of the chromosome. So just like a sweater is one sweater woven together by the tight stitching and weaving and knitting of the wool and yarn that makes it, this is one chromosome of DNA made up by the tight compacting weaving and knitting of the 30 nanometer fiber DNA that makes it. With all of this in mind, we can shift gears from thinking about DNA as a physical biochemical molecule to thinking of it as an information storing apparatus or structure. And by shifting our thinking from physical to information, we can begin thinking about how DNA is not only handled by the cell, but used by the cell in order to build proteins. So all you really have to do is take one look at DNA in order to realize that it can be and was evolved to be the heritable material of life. Watson and Crick, in their foundational paper, where they described DNA, and of course, as we all know, left out their major contributor as well, who should have gotten their Nobel Prize, Rosalind Franklin. Watson and Crick said it has not escaped our notice that the specific pairing we have postulated, they're referring to the adenine, thymine, cytosine, guanine base pairing, that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what they realized even then was because A always base pairs with T and C always base pairs with G, cells could and should know how to always copy their DNA using one existing strand as a template to build a second. In other words, if we pull DNA apart, and we previously had a cytosine guanine base pair, but now we pull those strands apart and we're only looking at a cytosine, we don't have to remember that a guanine was across from that cytosine before. We know it by the rules of base pairing. So fairly simple experiments done a very long time ago conclusively demonstrated that DNA was replicated in this semi-conservative way. One old strand is used as a template to build a new strand, and more importantly, that segments of DNA, genes between you and me, contain the information for building proteins. So here it is, the central dogma in all of its glory. We have DNA molecules, we have chromosomes, and those DNA molecules are double-stranded, anti-parallel. We have phosphate sugar backbones, here represented in orange, when we've got bases in the center held together by hydrogen bonds, and we can easily replicate that DNA by using the rules of base pairing. If we separate these two strands, because A always base pairs with T and G always base pairs with C, we only need one strand in order to deduce and make a second strand new. And that's great for DNA replication. But we can also separate these DNA strands and use them as template, leveraging the rules of base pairing, in order to copy specific genes into a secondary molecule called RNA, ribonucleic acid. And we do that through the process of transcription. Those RNA copies of DNA gene sequences can then be decoded or translated into amino acid chains, and that is our protein. And remember, proteins do everything. Everything. If we are thinking of life, we're thinking of proteins. You're sitting there because of proteins. You're breathing because of proteins. You're thinking because of proteins. You're alive because of proteins. And it pains me to say this to you. I'm near tears and have a lump in my throat because I'm a DNA person. I'm a DNA guy. I did all of my research is based on DNA. To me, DNA is the most marvelous thing in the world, and even I have to concede that proteins are responsible for everything we see. The only problem with proteins is they don't last forever. Like any good machine, they break down due to wear and tear. So when proteins break down, you've got two choices: die or make new proteins. And if you want to make new proteins, the question then becomes, how are you going to know how to make them? Where is your protein blueprint manual? And that's what the DNA is. So DNA is nothing more than a repository for making proteins. Proteins keep us alive. Problem is, we only know how to replace those proteins to keep being alive using the information encoded in DNA. For protein, structure equals function. The three-dimensional shape that proteins take dictate the function that they can do. And that three-dimensional shape of proteins is based exclusively on the amino acid sequence of those proteins. So if we want a protein to do a certain job, we need that protein to have a certain shape. If we need a protein to have a certain shape, that means we have to have that protein have a specific amino acid structure, sequence. And that sequence of amino acids is what the information in DNA encodes. So DNA tells the cell what the sequence of the amino acids needs to be in the protein. Once we concoct that sequence of amino acids, that protein will fold into the right three-dimensional shape. And with the right three-dimensional shape, it'll do the right job, and we get to live another day. In eukaryotic cells, one gene contains the instructions for building one protein. That's a little bit of an oversimplification, but that's a graduate level complexity, so we'll keep it there for this course. And if the need for a particular protein arises, either because that protein has broken down over time through wear and tear, or maybe we've just had an injury and we need an upsurge of repair protein so we can close a wound, for whatever reason we might need a protein, the region of DNA, the gene in the genome that contains the instructions for building that needed protein, is then activated. It's turned on. And the first step of being activated as a gene is that you're copied into an RNA molecule through transcription. Then that RNA molecule can be decoded to deduce an amino acid sequence, and that amino acid sequence will allow that protein to fold into the right shape, it'll have the right job, and the cell's need for that protein is satisfied. These RNA molecules that are made by transcription are nothing more than nucleic acid copies of the DNA gene. We've stayed in the same language of nucleic acids. The information hasn't been changed. That's why we call it transcription, because we've stayed in the language of nucleic acids. We're just copying or transcribing from one source to another. Only when we decode or translate those RNA sequences into amino acids, as we go to nucleic acids to amino acids, do we translate from one language to another. And now we've built a protein. We've used nucleic acids to deduce the sequence of amino acids. We've gone from one language to another, translation. Again, we call this information flow the central dogma, DNA to RNA to protein. And to have a little bit of a metaphor here or an analogy, if we want to think of the genome is the sum total of protein building information that the organism would ever need in its entire lifetime. We have in our genomes genes that we only needed as embryos and will never need again. Those genes are still in our genome now because we needed them at some point in our development. Those genes were very active during embryogenesis. They were shut down or silenced when we were born, and they'll never be activated again. But we have those genes in our genome because we needed them. So the genome, all of our chromosomes, is the sum total of every single protein we might ever need in the entire course of our lifetime. Just like an encyclopedia or a Wikipedia is the sum total of knowledge that we have. But at any given time, we only need a subset of that information. If we're writing a report for a history class on this town Wrexham that's in Great Britain, we only need that information from the encyclopedia. We might have all the information in the encyclopedia, but we're only accessing the specific information we need at that one time. And it's the same for our cells. Each gene is like an entry in the genome of the organism, and cells will access specific genes as they need those proteins to be built. So in this analogy, the encyclopedia is the entire genome. It's all of the DNA in the cell. It's everything, all the information you would ever need, but way too much information for any one time. And each entry in that genome and accessing those genes, copying them in a way that can be used and portable, is a messenger RNA. You can think of a messenger RNA as a Xerox copy of the page of the encyclopedia that you need. You don't have to carry the whole encyclopedia out the door with you. You take it to the photocopy machine, you make a photocopy of that one page, and now you have a portable, usable copy of the information you needed from the encyclopedia and nothing else. That's the mRNA, a short, portable sequence of nucleic acids that is small, usable. It's the information you need at that one time, and you can use it to achieve your outcome. Your outcome, of course, making the protein that the cell needs. So again, just to review the terminology so that you don't confuse them, students will often confuse the terms translation and transcription, and that's certainly very easy to do if you're memorizing these terms, and it's very, very hard to do if you understand why these terms were chosen. To transcribe something means to stay in the language, right? Medical transcriptionists, legal transcriptionists, if you hire a transcription service, you're sending them a video or an audio file of someone speaking English or Spanish or Chinese, and they are typing English or Spanish or Chinese. In transcription, you always stay in the same language. The only thing that changes is the form. You've gone from audible language to written language. That's transcription. And the same is true in a cell. In a cell, you are going to go from the language of nucleic acids to the language of nucleic acids in transcription. You're going from DNA, one form of nucleic acids, to RNA, a second form of nucleic acids, but you haven't changed the language. You've only changed the form. You have transcribed. Translation, of course, is completely different. In translation, you are switching languages. You're going from German to English, English to Spanish. You're switching the entire language, and the same is true for the term translation here. Now we're going from the language of nucleic acid to amino acid. We're going from RNA to protein. That's a complete language shift, and it requires a much more laborious process. So the language of DNA and RNA is the same. It's the language of nucleic acids, of nucleotides. DNA is very similar to RNA. They're both linear polymers. They're both made up of 4 nucleotides. They're both held together in chains with phosphate sugar backbones. They're both 5' to 3'. There's actually only a few differences between DNA and RNA, and they're almost like accents, right? I was raised in Brooklyn, so I say dog, not dog. I always make fun of my family because they say dog. I say dog. I say ball, not ball, or however you all say it. I say ball. So those are accents. I'm speaking English, you're speaking English, but you say ball, and I don't know what you say. I say ball, and you say ball, however you say ball, but we say it differently. Same is true for DNA and RNA. DNA has one hydroxyl group. It is on the 3' carbon. RNA has 2 hydroxyl groups, one on the 3' carbon and one on the 2' carbon. So by looking at this chain here, I know right away that this is RNA, because in DNA, this would just be a proton. This would just be an H. But here we have this hydroxyl group on the 2' carbon. That's only true for RNA. Additionally, there's no thymine in RNA. RNA doesn't have T's. It has uracils instead. Again, almost like an accent, right? I always joke with students that RNA doesn't know how to say T. It says U instead. Every time there's an adenine in RNA, there's a uracil base paired to it if there's base pairing going on. So uracil does the same job as thymine, but uracil takes the place of thymine in RNA. And then finally, most of the time, RNA is usually single-stranded. RNA can base pair within itself if the chain of RNA is long enough. We can have some 3-dimensional folding of RNA, but RNA doesn't typically exist in 2 double-stranded molecules, 2 antiparallel strands coming together the way that DNA does, which makes it much more adaptable, but it also makes it much more fragile. So here's some examples of the intramolecular base pairing that's possible in RNA. Here's the 5' end or beginning of this RNA molecule, and if we trace it, we can see that this really is one long single chain of RNA. This is one strand of RNA, but because it's so long, it can base pair within itself. We've got a region of complementarity here, so the RNA is base pairing with itself here and here, here and here, here, so we can have this RNA kind of take on this shape. And here we see a little bit more of an accurate space-filling molecule of the same thing. So RNA can be double-stranded, but it's usually not double-stranded with another separate strand of RNA. It's double-stranded within itself. DNA can't do this. DNA is double-stranded with partner molecules. So again, we've got DNA as an information storage repository. We've got RNA as a means to make short portable copies of the information that DNA encodes. And then we can make protein from that RNA. Information storing, very stable, very static in the DNA form. Proteins are very, very catalytic, right? They are dynamic. They cause chemical reactions to occur. They move. They contain no information at all. Proteins are doers, not the knowers. And the RNA is the intermediate. It's somewhere in between the two. RNA stores information for sure, because it carries that information to translation, but it's not very stable because it's single-stranded. And RNA can be catalytic in its double-stranded intramolecular base-paired form. When RNA takes on 3 dimensions, it can be catalytic, but it's not very good at catalysis. So it stores information like DNA, but not so well because it's fragile. It performs catalysis like protein, but not so well because it's not completely 3-dimensional. And so RNA is the intermediate. There's not to digress too much, but there's a theory called the RNA world hypothesis that states that the first biomolecule that evolved on this planet was most likely RNA. And then RNA evolved in 2 branched directions. It evolved to become DNA to optimize its information storing capacity, and RNA also evolved to protein to get very, very good at catalysis. That's what allowed life to thrive on this planet. You had DNA molecules dedicated to information storage and heterability. You have proteins dedicated to biochemistry and catalysis. But because those 2 classes of molecules were so different, DNA and protein, RNA persisted as the intermediate, the go-between between these 2 forms, because it is the overall ancestor of those 2 biomolecules. So, basis of all life really comes down to RNA, which is interesting in itself. Again, this is the central dogma, the overarching pillar of knowledge of all of molecular biology, the idea of information transfer from DNA to protein. So we're more or less halfway through the lecture. As I said, it's going to be a long one. But now we're going to shift gears completely to gene expression. We're going to talk about the central dogma in detail. How does transcription work? How does translation work? How do we make proteins from DNA genes? So again, transcription produces an RNA complement or an RNA copy of a DNA gene, a DNA strand. Transcription is the only way that a cell can make RNA. Anytime RNA is made from DNA in a cell, it's being made through a transcriptive process. The process has to start with the unwinding of the DNA compaction and the opening or the separating of the double strands, making those 2 strands accessible. We need to make DNA single-stranded, at least in the region of the gene, so that the transcriptional proteins, the machinery, the enzymes for transcription can access that DNA. And we usually begin that separation just in front of a gene. We use the terms upstream or 5' of the gene, meaning right before it. So this is actually a bacterial diagram. We don't have sigma factor in our cells, but if the gene is right here in this DNA, or maybe it's right here on the right, we would begin opening those 2 strands, making a replication, a transcription bubble just at the beginning of the gene, just at its start, so that we can begin accessing that DNA. And we do this to free up the bases so that base pairing is no longer in place, so that we have a bare cytosine and we know a guanine goes across from it. We have a bare adenine and we know a ... uracil goes across from it. Remember, we're making RNA here, so we put a uracil in across from an adenine. And that's what we see happening here in detail. So in blue are the DNA strands. They've been pulled apart to this transcription bubble. We have the RNA being made by the enzyme RNA polymerase. The RNA is in red. And we see we're following the rules of base pairing. We're using this bottom DNA strand as a template in order to make a new strand of RNA. Cytosine across from guanine, adenine across from thymine, uracil across from adenine, and guanine across from cytosine. Those ribonucleotides, the building blocks of RNA, are called NTPs, not dNTPs. Those ribonucleotides are added one by one to form a chain, 5' to 3', and that chain is going to be complementary to the template strand on the DNA. It's going to match it in base pairings. We can refer to that newly formed mRNA molecule as a transcript because it was born from the process of transcription. And again, we do have some differences between transcription and replication. The newly made RNA strand doesn't stay base paired with the DNA. As soon as transcription terminates, that RNA is going to float away and be used for translation. And immediately behind this transcription bubble, immediately behind the RNA polymerase enzyme, the DNA is closing up again, going back to being double-stranded. So here again is RNA polymerase in light blue this time. The DNA is in gold. Here is the process of transcription occurring in real time. We're getting that base pairing between the template bottom strand of DNA and the newly made strand of RNA. But here we see the RNA we've already built is actually exiting this process through a specific channel. It's not getting in the way of this DNA re-annealing, re-base pairing here behind the RNA polymerase. We've said this already, but RNA copies, their sole point is to be portable and usable. That means they're smaller than DNA, much shorter than DNA. They're only copying the gene that we want to express, not the entire chromosome. And it's being done by RNA polymerases. RNA polymerases, as an enzyme, they catalyze or oversee the formation of new phosphodiester bonds, new bonds between the NTPs or nucleotides of RNA, building that phosphate sugar backbone. So this is RNA again. I know it's RNA because I have a uracil here, and we can see that we've got this sugar, phosphate, sugar, phosphate, sugar, phosphate backbone, and it's these particular bonds that we see indicated here, these phosphodiester bonds, that are being catalyzed by RNA polymerase. This is one nucleotide, and it's being linked to the next nucleotide through this bond right here. Each time one of those phosphate bonds in a nucleotide is broken, a lot of energy is released. It's that energy that powers this process of transcription. RNA is only associated with template DNA in a very small region, in the region where transcription is actually occurring, in that transcription bubble. And so we don't really take up a lot of room with this process. We can have multiple RNA polymerases transcribing a gene simultaneously. They kind of march along like garbage trucks or like soldiers in tandem, one right after the other, transcribing in this wave. And we see that here. In this case, the RNA polymerases would be going to the left. So this is the 5' end of the gene. This is the 3' end of the gene. And we see the RNAs are going in this direction here, transcribing from the beginning to the end. And as they're doing so, they're making this red RNA molecule. This one is the furthest along, this RNA polymerase, so it's got the longest messenger RNA. It's actually approaching the termination site, and there's a few behind it. So we can make a lot of messenger RNAs at any one time. This is an electron micrograph of that same process. You can see that lots and lots of RNA polymerases are transcribing a gene at any one time. These are two genes that we see in the field of view. This is non-coding DNA between these two genes. You might be able to just see this finest of fine threads right here between these two genes. And we can see that these genes are being actively transcribed by many, many RNA polymerases. This is the beginning of the gene. The RNA polymerases here are going left to right. These are the RNA polymerases that have gone the furthest, and they've got the longest messenger RNAs coming off of them. And these are the ones that have gone the shortest distance, and their mRNAs are still in process. They're still much shorter. Same as the truth here, beginning of the gene, end of the gene. So I'm not going to count these either, but what would you guess? Maybe 60, 70 different transcripts being made at any one time? So a lot of transcription occurring at genes simultaneously. Transcription is pretty slow. We can add 30 bases of RNA per second, but we make up for that slow speed by having this simultaneous transcription, being able to make many messenger RNAs at any one time. And I don't know how many of you have learned about DNA replication in other courses, but when we replicate DNA, we always need to start with a primer. That's not true for RNA. So no primer needed for RNA synthesis, even for its initiation. RNA polymerase can land on a regular gene and just start transcribing from scratch. Pretty high error rate in RNA polymerase. We make an error in 1 out of every 10,000 bases transcribed. We have a mistake. So low, but much, much higher than DNA replication. That higher rate of error is tolerated because RNAs don't last for long. They're usually lasting on the order of minutes to at most weeks. So even if we have a mutant RNA, it's going to be degraded over time. We'll be making new RNAs behind it, and they won't have that mistake in it. So we don't worry too much about mutations in RNA. Certainly there's no RNA repair mechanism because the mutant proteins aren't going to last that long. All right. So, how do we start this transcription process? How do we initiate transcription in eukaryotic cells? Transcription initiation in our cells is fairly complicated because we are more complex cells in general. But let's highlight some differences from bacteria, some things that make us different from bacteria. First of all, human cells, all eukaryotic cells, have 3 RNA polymerases. RNA polymerase 1, 2, and 3. Bacterial cells have only 1 main RNA polymerase. RNA polymerase 1 in our cells is responsible for transcribing ribosomal RNA genes. RNA polymerase 3 transcribes transfer RNA genes, smaller ribosomal RNA genes, and some small microRNAs. RNA polymerase 2 is the one we're going to talk about today. RNA polymerase 2 transcribes protein coding genes, the genes that are the instructions for building proteins, and that's really the primary feature of transcription. Our RNA polymerase cannot start transcription unaided. It needs a landing area called a promoter. It also needs additional proteins to start the process of transcription. In bacteria, transcription can start with just an RNA polymerase alone, sometime. The proteins that eukaryotic cells need to start transcription are called general transcription factors, or GTFs, and transcription cannot occur in a eukaryotic cell without these GTFs. GTFs assemble at the gene promoter, at the starting point of the gene, the beginning of the gene. They recruit RNA polymerase 2, and together, RNA polymerase 2 and these GTFs form a pre-initiation complex, or a PIC, a PIC. Since the process is more complex in eukaryotic cells, it's also highly regulated, and we'll talk a little bit about the regulation of gene expression in later lectures. Finally, there's no chromatin or DNA compaction in bacterial cells. DNA in bacterial cells is compacted simply by wrapping it, coiling it very tightly, something called supercoiling. There's no nucleosomes, there's no histones. So in order to transcribe DNA and to express genes in eukaryotic cells, those eukaryotic cells have to deal with chromatin. And that's also something we'll discuss in some later lectures. So let's talk more about these general transcription factors. Again, they're responsible for setting the stage at the gene promoter so that the RNA polymerase enzyme can be recruited to that promoter and so that it can initiate transcription. So the GTFs not only recruit RNA polymerase, but they position it in three-dimensional space correctly, and they begin the process of separating double-stranded DNA into a transcription bubble so that transcription can occur. All of the GTFs are named in a very similar way to one another. They all start with a TF, that stands for transcription factor, a Roman numeral that determines which RNA polymerase it associates with. Since we'll only be talking about RNA polymerase 2 amongst ourselves here, we'll only be talking about TF2s. The transcription factors for RNA polymerase 1 and RNA polymerase 3 are called TF1 and TF3. TF2 is the transcription factors for RNA polymerase 2. And then finally, each individual GTF just gets an arbitrary letter. So we'll be talking about TF2D, TF2H, TF2E, TF for transcription factor, 2 because it's associated with RNA polymerase 2, and then the last letter is just the arbitrary unique identifier for each individual GTF. For RNA polymerase 2, the first GTF to recognize and bind the gene, the first GTF that physically binds to the promoter of a gene that needs to be transcribed, is TF2D. TF2D contains as one of its subunit proteins a protein called Tata binding protein, TBP. And Tata binding protein binds to a sequence of DNA called the Tata box, And the top box is the gene promoter for eukaryotic genes. So every single gene, well, that's an oversimplification too, not entirely accurate, but for the sake of simplicity, every single gene in the human genome starts with a TATA box. That's the on-off switch of the gene. It is the gene's promoter, and TATA binding protein binds to that promoter, TBP. TATA binding protein binds to the promoter. That promoter sequence is very, very rich in T's and A's. We might come back to that later in a challenge question. And it's found about 25 bases in front of or upstream of the very beginning of the coding region of the gene. And again, we call it the TATA box. When TBP binds to that promoter region, when TBP binds to the TATA box, it kinks the DNA about 90 degrees, and that kink marks that region as a region the cell should be actively transcribing. This makes the promoter unmistakable, and the other GTFs begin to recruit to that kinked DNA, and they'll eventually position RNA polymerase II there as well. So the very next GTF that binds after D is TF2A, and then TF2A, and then TF2B comes in. After TF2B, TF2F comes in, and it escorts the RNA polymerase II itself. That's F's job, is to bring in RNA polymerase II. And then E, and then finally H. So we use the acronym DABFA to represent this order of GTF recruitment to eukaryotic genes. DABFA, 2 syllables. And what DABFA tells us is that the very first GTF to bind to the promoter is TF2D, D for DAB. And after the D comes TF2A, and then TF2B. And then we've got that break, right? That syllable break, DAB-FA. So that break means that TF2F is bringing in the RNA polymerase as well. And then TF2E and TF2H. So this is the order of recruitment of the GTFs. The TF2H does a very specific job. It's perhaps one of the most important GTFs after TF2D. Much like the histone proteins, RNA polymerase II also has a tail, a carboxy terminal end of itself. And that carboxy terminal tail, that last few amino acids that projects out of the central core of RNA polymerase II, is functional. It's accessible, and it can be modified. This is the carboxy terminal end of the protein, not the amino terminal end. So this really is the end, the bottom, the tail end of the protein. This carboxy terminal tail of RNA polymerase II can be phosphorylated. It can have phosphate groups added to it, and that phosphorylation serves as a type of code, an RNA polymerase code. Based on those phosphorylation patterns, the cell and the enzyme know what stage of transcription it's in. For example, completely unphosphorylated RNA polymerase II, non-phosphorylated RNA polymerase II, is involved in assembling with the GTFs. It's going to be at the promoter. It's not transcriptionally active. When we phosphorylate serine 5 in the carboxy terminal tail, that is the initiating form of RNA polymerase II. It has just begun active transcription. When serine 2 and serine 5 are both phosphorylated, now that RNA polymerase is in what we call the elongation phase. We are actively transcribing the gene. We're in the middle of the gene. And then when we dephosphorylate serine 5, so only serine 2 remains phosphorylated, this is an RNA polymerase II that's prone for termination. It's near the end, and it's going to soon be terminating transcription. Finally, that second phosphate is stripped off, so we have an unphosphorylated form of the RNA polymerase. This is after termination, and that unphosphorylated form of RNA polymerase is now ready to rejoin another promoter and be involved in recruitment and pre-initiation once again. So these phosphorylation signals control the state and function of RNA polymerase, and again tell the cell the status of that gene's transcription. Once RNA polymerase II is successfully transcribing, once we've transitioned to initiating and certainly to elongation, most of the GTFs dissociate from the promoter, and they're ready to assist transcription initiation again. Oh, and the one thing I negated to say, even though it's on the slide, I did say that TF2H is perhaps the second most important transcription factor, and it's because TF2H is the kinase, is the enzyme that's responsible for making these phosphorylation events occur. It phosphorylates that tail. Part of recycling RNA polymerase II, so it's ready to rejoin a new promoter, is stripping of all of its phosphate groups using a phosphatase enzyme. Only unphosphorylated RNA polymerase II can be recruited to a promoter. All right, so we've made a messenger RNA. We have transcribed a gene into an RNA. We have recruited RNA polymerase to our promoter using the DAB-Phe GTFs. We have phosphorylated RNA polymerase II so it could leave the promoter and elongate transcription. We've terminated that transcription by phosphorylating RNA polymerase only at serine 2. We've made a messenger RNA. Now we have to process that messenger RNA so that it's ready for translation. So just because the mRNA is transcribed doesn't mean we're ready for translation. It has to be quality controlled. It has to be modified, checked, and processed. For bacteria, that's simple. Newly transcribed finished messenger RNAs really are ready for translation, but not so in eukaryotes. In eukaryotes, we've got compartmentalization because of the nucleus, and we also have more complexity, which means we need a tighter regulation system. We need more monitoring. So the mRNA that was just made in the nucleus has to be quality controlled, modified, and exported from the nucleus in order for translation to occur in the cytoplasm. And that's what we're going to talk about now, mRNA processing. So we're going to inspect these messenger RNAs, make sure that they're whole, make sure that they have high integrity, and then process them to mark them as ready for translation in the cytoplasm. So really, mRNA processing breaks down into three main categories. Something is added to the mRNA's beginning, to its 5' end. Something is added to the mRNA's end, its 3' end. And something is done to the center of the messenger RNA. And only mRNAs with those three modifications can leave the nucleus for translation. The enzymes responsible for these modifications actually ride along with RNA polymerase II. They actually interact with the RNA polymerase tail, that CTD tail, and they are making these modifications and making those checks for mRNA integrity as transcription is ongoing. So we've got capping factors, polyadenylation factors, and splicing factors, and we'll talk about all of that in just a second. So the very first thing that occurs to a messenger RNA is that it gets a 5' cap. And this 5' cap is really just a guanine nucleotide, but it's added to the 5' end of the messenger RNA in an inverted way. It's upside down relative to the other bases. We can see that here. And it's methylated. So this methylated upside down guanine is added to the 5' end of the messenger RNA, and it marks that 5' end as genuine, as real, as quality controlled and quality assured. We also add to the very end, 3' end of the messenger RNA, a tail. It's a poly-A tail. It's usually a couple of 100 of adenines long. It's just a run of straight adenines added to the 3' end of the messenger RNA. And that marks the 3' end as real, genuine, and quality controlled. This polyadenylation requires a couple of different enzymes. One to actually chop the messenger RNA off of the elongating polymerase II, and then other enzymes to add the poly-A tail. And again, there's quality control measure. These two modifications result in the messenger RNA being more stable. These modifications also mark the messenger RNA as a messenger RNA molecule. Anything with a upside-down methylated guanine cap and a poly-A tail is a messenger RNA, and the cell knows that that should be translated. In other words, these modifications yell out to the cell, hey, I'm something to make a protein from. Come and translate me. That should say, come translate me, not transcribe me. Come and translate me because I am ready to be decoded. Finally, proteins are recruited to the 5' cap and the poly-A tail, and those proteins interact with one another. They form this circularization of the messenger RNA. This is called a cap-binding protein, and this is a poly-A binding protein here. And cap-binding protein and poly-A binding protein interact with one another to circularize the messenger RNA. Any circularized messenger RNA tells the cell that that messenger RNA is whole. In other words, there's no fracture or break in the messenger RNA in the middle of the sequence. That means you have an intact cap, an intact tail, and that cap and tail are still connected on one single messenger RNA molecule, making it a circle. So circularized messenger RNAs are what are translated in the cytoplasm. Any linear messenger RNAs indicate that a breakage occurred somewhere in the molecule. It's not whole anymore. But we're still not done. We've got that one last processing to do. Most eukaryotic messenger RNAs are also chopped up and stitched back together. As odd as it sounds, that's what happens. Most eukaryotic messenger RNAs are actually interrupted by non-coding sequences that do not contain information for building proteins. These non-coding sequences get in the way of the coding regions, and they are disruptive. Without going too much into the history of it, this wasn't on purpose, this wasn't evolved intentionally. Most of these interrupting sequences are actually viral remnants. These are scars in our genome from old historic viral infections where the viruses have mutated and are no longer active in our genome, but their viral DNA is still there. So we got to get all that crap out of the gene so that we have one continuous protein coding sequence in the messenger RNA. So almost all of our genes in eukaryotic cells have their protein coding regions interrupted by these viral remnants, by these non-coding interrupting sequences, and we call those introns. The coding regions that we want, the information of the gene that actually does have the protein coding information, are called exons. So we want to keep the exons and stitch them together and chop out and remove the introns because they're in the way. And only when we do that will we have a continuous coding region that contains only protein coding information. Introns can range anywhere from being very, very small, a single nucleotide, to being very, very long, tens of thousands of nucleotides long. And we got to get them out of there. We got to keep the exons only. For most genes, there are more introns than exons, but doesn't matter. We still got to get rid of them, and we do that through a process called splicing. So once the mRNA has been transcribed, and that transcribed pre-messenger RNA will contain both introns and exons from the DNA, we have to do this splicing to remove the introns before that RNA can be translated. So functional, all-coding, mature messenger RNAs are made through this process called splicing, where the introns are removed and the exons are stitched back together. So here we see a very long intron in orange. We need to cut that intron out and join these blue exons together so that everything is protein coding. RNA splicing is a very elegant, quite complex process that we'll just briefly introduce here. We won't talk too much about how it works, just give you a sense of how introns are spliced. Each intron does contain some limited conserved sequences that all introns have in common, and those sequences tend to be near the ends and in the middle of the intron. So cells can recognize the beginning, the end, and the center of an intron, and these sequences serve as a cue to remove those introns. Using these sequences as a guide, a very complex protein machine called a spliceosome will cut the intron at its 5' end, join that free 5' end to the center of the intron, cut the intron at the 3' end, and then remove the intron. This is the graduate level mechanism of the spliceosome. We won't get into the spliceosome in that way at all. All that we need to know is that when we cut the intron at the 5' end and we join that to the middle, we're actually joining that 5' end of the intron to an adenine in the middle of the intron, and that forms what we call a lariat. So here's the image we want to know for this class. Again, the exon is in blue, the intron here is in yellow. The spliceosome is going to first cut the 5' end of the intron. It's going to free that 5' end, and it's going to join that 5' end to an adenine in the center of the intron. We see that adenine here. That forms this lariat structure. Once the lariat is formed, we're going to cut the intron at its 3' end, and that frees the entire intron as a lariat, and then the two exons are joined together, as we see here. The lariat floats off, and it's eventually degraded. Its individual nucleotides are recycled for future rounds of transcription. And that's it for mRNA processing. Transcription was the easy part, right? We separated DNA into single strands. We copied one strand of DNA into an RNA strand. If there's a thymine in the DNA, we put a uracil in the RNA. But other than that, pretty straightforward. Once transcription is done, the messenger RNAs need to be checked. Make sure they're intact. If they're intact, they get the 5' cap, that upside-down methylated guanine. They get their poly A tail, and they get spliced with the spliceosome. because the cap has cap binding proteins associated with it and the tail has tail binding proteins or poly A binding proteins associated with it, that messenger RNA can circularize, and circularized messenger RNAs can then leave the nucleus. So only messenger RNAs that make it through all three of these mRNA processing events can leave the nucleus as circles for translation. And that allows the cell to do some really extensive quality control. Transport out of the nucleus occurs through nuclear pore complexes, which you can learn about in a cell biology course. Nuclear pore complexes only allow messenger RNAs through that are capped, tailed, and contain splice junctions. And we monitor that through these binding proteins. So only proteins with poly A binding, only messenger RNAs with poly A binding proteins, with cap binding proteins, and with these exon junction complexes, these splice junction proteins, show the cell that all three of those processes occurred, that messenger RNA is quality controlled, and it can leave the nucleus for translation. So these proteins, just by their mere presence on the messenger RNA, certify that the messenger RNA has been capped, tailed, and spliced, and is ready for translation. Any messenger RNA present in the nucleus that doesn't contain all of these three proteins fails the quality control check, and it will be recycled for its basic building blocks. So now we are ready to move on to the last topic of this lecture, translation. How do we take that intact, complete messenger RNA and translate it or decode it into a chain of amino acids? So again, conceptually, transcription is the easier of the processes we're talking about today. There's not much to it, right? If you understand base pairing, you understand transcription. A's go with U's because it's RNA, T's go with A's, G's and C's go together. You separate the DNA, you have a single strand of DNA, you can copy it into RNA. The translation is different because we're staying not in the same language of nucleic acids, but we're switching languages. How do you get from nucleic acids to amino acids? How do you translate from a language of 4 letters, C's, T's, A's, and G's, to a language of 20 letters because we have 20 amino acids that make proteins? That's a debate that raged in biology for quite a while until it was all figured out, but now, of course, we know exactly how it works, so let's dive in. First thing is that lack of correlation in numbers of letters really is the first problem, translating from 4 nucleotides to 20 amino acids. How do you get a language of 4 words or 4 letters to translate into a language of 20 words or 20 letters? It's not straightforward. We can think of other languages that are based on symbols, that are based on single information containing units, and we know that the translation between languages of that type can be quite challenging. But let's see how it could work. Obviously, if we go to a one-to-one relationship, it's not going to work at all. If one nucleotide coded for 1 amino acid, we could only capture the information for 4 amino acids, right? In other words, if cytosine encoded serine, if thymine encoded lysine, adenine encoded glutamic acid, and if the fourth nucleic acid went with a 4th amino acid, what about the other 16 amino acids? How are we going to code for them? So that doesn't work. If we have 2 nucleotides coding for amino acids, that would give us 16 possible combinations. 4 times 4 is 16 different combinations of 2 nucleotides. AA, AT, AG, AC, TA, TT, TG, TC. We got 16 of those. So we could have 2 nucleotides encoding each amino acid, each single amino acid, but still we'd have 4 missing. We've got 20 amino acids. We have only 16 combinations of 2 nucleotides. We need 20. So we got to go to 3 nucleotides encoding each amino acid. 3 nucleotides gives us 64 possible combinations. AAA, AAT, AGG, AGC. We get 64 unique combinations of 3 nucleotides, and we only have to encode 20 amino acids, so at least we can do it there. And that's what we evolved. So each single amino acid is encoded by 3 nucleotides. 3 nucleotides makes up the word of a single amino acid. And we only need 20, so 64 is overkill, but that's actually going to come into be some use, as we'll see in just a second. So these sets of 3 nucleotides at the DNA or RNA level that contain the information for a single amino acid are called triplets or codons. We can use those 2 terms interchangeably. And if 3 DNA or RNA triplets contains the information for a single amino acid, then we can deduce a nucleic acid to amino acid dictionary. We can truly translate. And we have done that. We call that the genetic code. And here it is in all of its glory. This is the genetic code for RNA. Right? So in the center here, we have all of those 64 codons, all of those 64 triplets, the 64 different sets of 3 nucleotides that we can have. And we also see what amino acids those codons code for. The way we use this table is if we have a specific codon we're looking for, let's just say ACC. The first letter is A, so I'm going to restrict my looking into this row. The second letter is C, so I'm going to come down here where those two intersect. Now I have AC. All the AC codons are in here, and then I can use the third letter here, ACC. That's this codon right here. ACC codes for threonine, THR. And you don't have to know what the 3-letter codes code for. You don't have to memorize what they code for. You could just say THR. But that's threonine. So this is our nucleic acid to amino acid dictionary. This shows us what each of the codons codes for. Now, what you might have noticed in the previous slide in the genetic code is that because we have that overkill, because we have 64 different possible combinations and only 20 amino acids that we need to encode, we have the opportunity for redundancy. So leucine is encoded by 6 different codons, UUA, UUG, CUU, CUC. All of these codons encode leucine. So leucine is what matters in the gene. If we do have a mutation where the CTC codon at the DNA level mutates from CTC to CTT, well, the codon would go from CUC to CUU, but leucine is still encoded there. So it really doesn't matter for the protein, right? Protein would be exactly the same, and protein is all that matters because the protein does everything. So this redundancy gives us some tolerance to mutations. So because of this redundancy, we can have quite a few mutations that we refer to as silent because they don't change the amino acid we're encoding at all. So a little bit of a buffer or protection against mutations. Since it's all about the proteins, we can have a horrible mutation that's really not horrible at all because the protein coding hasn't changed at all. All that matters are the amino acids inserted into the protein. If those amino acids are unchanged, the protein is unchanged. So with a few very subtle, very, very small number of exceptions, all life on this planet uses this genetic code for its translation. We are all unified through evolution, and this is the universal genetic code for all information, all life on Earth, including chloroplasts, mitochondria, and single-celled bacteria. So that's great in theory, right? We've got codons in the messenger RNA. We've got amino acids that those codons encode. So abstractly, I'm beginning to understand it. CUC is leucine. So if there's a CUC in the DNA gene, or CTC, in the DNA gene, and CUC is in the messenger RNA, the cell knows to put leucine there in the protein. But how? Physically, how? How is leucine bridged to the messenger RNA? And that's where tRNAs come in. Transfer RNAs are the true translators of this process. The codons in the messenger RNAs don't directly bind to their corresponding amino acids. CUC doesn't recruit leucine for translation. Instead, we need adapter molecules to bridge that CUC codon to a leucine, and those adapter molecules are transfer RNAs. Transfer RNAs have a 3-nucleotide anticodon on one end that's complementary to the codon. So here we see the codon is GCC, and GCC encodes alanine. If you don't believe me, we'll just go back. GCC, here's GCC, it encodes alanine. So here's the GCC codon at the DNA, at the RNA level. And a transfer RNA is base pairing with that. Transfer RNA has the anticodon GGC, which is base pairing complementarily with the GCC codon. And that transfer RNA is going to have on its other end alanine, and it's going to bridge that alanine amino acid to the codon. So the anticodon of the transfer RNA directly binds to, using base pairing, the codon on the messenger RNA, and it carries with it the amino acid that that codon encodes. Again, the T in tRNA stands for transfer, but I always think of that T really standing for translator, because in my mind, it is the tRNA that speaks both languages. Only the tRNA has both nucleic acid information on one end and amino acid information on the other. Only transfer RNAs, only tRNAs, speak both languages, so again, I think of that T as standing for translator RNA. It's the only molecule that bridges those two worlds. tRNAs are one of those self-base pairing molecules we talked about earlier. It's a single strand, each one is a single strand of RNA, but it has intramolecular base pairing. About 80 nucleotides long, but it base pairs within itself quite a bit. And there are 4 regions of extensive base pairing that forms what we call this cloverleaf structure. So here's the beginning of the tRNA. We can see even at its beginning it's base paired into this stem. And then we go into a second stem here. Here's a loop region in orange. There's the rest of the complementarity for the second stem. We go into a third stem here, another loop in blue. This is the anticodon loop. This is where the anticodon is found. And then we go to the second part of that intramolecular base pairing. Here's our 4th stem loop structure. And then the very end of the tRNA completes the base pair, so that first stem is base paired. And at the very 3' end, we have the amino acid attached. This is showing a AAG anticodon. Again, we always read DNA. Oh, I'm sorry. This is showing a GAA anticodon. We always read our RNA 3' to 5' to 3'. So GAA is going to base pair to a sequence that is UUC, right? Because we always have base pairing that's anti-parallel. So if this is GAA in the 5' direction, it's going to base pair with something UUC in the 3' direction. UUC, if this image is correct, should be the codon for phenylalanine. And let's check. UUC, phenylalanine. UUC, I didn't plan this, so I hope it's true. UUC, and there's phenylalanine right there. So again, proof to the pudding. This is the correct tRNA for phenylalanine. It recognizes the phenylalanine codon on its anticodon end, and it's carrying phenylalanine on its other end. This is in 2 dimensions. This cloverleaf is then going to fold in half into 3 dimensions and yield a further complex, more L-shaped structure, but the idea is still the same. We've got the anticodon here in blue, and we've got phenylalanine on the other end. And this is going to be the bridge for translation. It's going to link the codon on the messenger RNA to the amino acid in the protein. Even when it's in this configuration, the 2 most important parts of the tRNA are its 2 ends, where it's recognizing codons in the messenger RNA and where it's carrying amino acids for translation. Unifying all of this and coordinating all of this is the ribosome. The ribosome is going to hold the messenger RNA and hold the tRNAs and guide the amino acids into building a chain, a protein chain of amino acids. The ribosomes are going to oversee and coordinate translation. Much like the spliceosome, ribosomes are huge, huge complexes of protein and RNA. And again, their single responsibility is to coordinate translation. Made up of dozens and dozens of proteins, nearly 100 different protein subunits, and 4 catalytic RNAs come together to make the 2 main subunits of the ribosome, a large subunit and a small subunit. And we'll see in just a second the messenger RNA is sandwiched between these subunits for translation to occur. These are really, really big. Eukaryotic and prokaryotic ribosomes are extremely similar in both their structure as well as how they coordinate translation. We first learned about this process as a scientific community using prokaryotes, and then we were amazed and shocked as scientists at how closely it mapped to what goes on in our own cells. This starts with the small subunit of the ribosome. The small subunit of the ribosome matches the mRNA codons with their proper tRNA. Small subunit facilitates codon recognition, and the larger subunit guides those tRNAs into the right place so that the amino acids they carry can be linked together into a peptide chain. Each amino acid is linked together using a peptide bond. The mRNA is pulled through the ribosome, much like a strip of tape from the tape roll, and as it moves through, these codons in the messenger RNA transit through 3 different sites in the ribosome, an A site, a P site, and an E site, and different things happen in each of that site to allow translation to occur. In the A site, new tRNAs enter the ribosome, bringing their amino acids with them. In the P site, those amino acids are linked to the growing chain of amino acids that was already in place from previous codons. And then the E site is the exit site. That's where empty tRNAs, now deprived of their amino acids, leave the ribosome. And the mRNA is decoded, one codon at a time, one amino acid at a time, with the tRNAs acting as the adapters, bringing in the right amino acid for each next codon. So again, as we go through this, keep in mind why tRNAs are the translator RNAs, why they are the bridge between nucleic acids and amino acids in this process. Ribosomes can add 2 amino acids to a growing peptide chain per second, so quite slow in the grand scheme of things. Bacterial ribosomes are quite a bit faster, but still impressive that this can be done at all. This process continues until a stop codon is reached. Stop codon, I don't think I have another genetic code here, but we have 3 stop codons in the genetic code. They are UAG, UAA, and UGA. So UAG, UAA, and UGA, 3 stop codons, and any one of those stop codons will terminate this process of translation through translation termination. So, let's kind of see this schematically rather than in words and see how this process all occurs. Again, the 2 subunits of the ribosome come together to sandwich the messenger RNA in the middle. Process of translation initiation, which we'll touch on a little bit later, just as its own unique process, is assuring that the messenger RNA is lined up correctly within the ribosome and that translation starts appropriately. In the larger subunit, there are those 3 voids that I talked about, those 3 sites, the P site, the A site, and the E site, and each is shaped perfectly to fit a tRNA. The A of the A site stands for aminoacyl because the A site accepts tRNAs carrying only one amino acid. The P in P site stands for peptidyl because this is where the tRNAs carrying peptide chains will reside. And the E site again stands for exit. It's where empty tRNAs leave the ribosome. Aminoacyl tRNAs carry only one amino acid, and peptidyl tRNAs are carrying the chain. And again, the name of the site tells you the kind of tRNA that is in that site at any one time. So let's watch it happen. First, the ribosome assembles around the messenger RNA, near the messenger RNA's 5' end, and then in a stepwise repeating fashion, translation is accomplished codon by codon, amino acid by amino acid, and then we get to termination. So let's pick things up in the middle, and then we'll fill in the gaps of initiation a little bit later, because it's its own complex story. So first, the tRNA with the correct anticodon carrying the correct amino acid is going to enter the A site, because any tRNA with one single amino acid can enter the A site, and it's going to bind to the appropriate codon with base pairing. The amino acid on this tRNA is going to form a peptide bond with the chain of amino acids that's being held by the tRNA in the P site, and that's going to transfer that whole chain to the A site tRNA. Now, tRNAs carrying chains of amino acids can exist in the A site. A site is only for tRNAs with single amino acids. So the whole ribosome is going to shift forward by one codon to get that new tRNA that's in the A site, carrying a peptide, into the P site where it belongs. And then that will leave the A site open for the next round. This is called translocation, where the ribosome moves forward by a single codon, moving the tRNA that was in the P site into the E site, moving the tRNA that was in the A site to the P site, and now the A site is vacant to allow the next tRNA to come in. Only 2 tRNAs can be in the ribosome together at any one time, so when a new tRNA enters the A site, the empty tRNA in the E site will be bounced out, and the process will continue. So here we see it in pictures, right? Let's describe where we are in context right now and watch a round of translation occur. So this is the empty tRNA. This is a tRNA with no amino acid on it at all, and it has just been evicted from the E site. It's been kicked out of the E site because we can only have 2 tRNAs in the ribosome at any one time, and these 2 sites, the A site and the P site, are both occupied. So because these 2 sites are occupied, the empty tRNA has been kicked out of the E site. Everything is fine right now. This is our peptidyl tRNA. It's a tRNA linked to a chain or peptide, a chain of amino acids or a peptide. So any chain of more than one amino acid, 2 amino acids, 3, 4, a dozen, 500, any chain of more than one amino acid is a peptide chain. And here we have a peptide chain linked to a peptidyl tRNA in the P site. Everything is good. And here's our newly bound tRNA in the A site. The A site is for aminoacyl tRNAs. tRNAs carrying only one amino acid, and that's what this is. So that's good too. Now, with the A and P site filled, the ribosome is going to catalyze the formation of a new peptide bond. See, this peptide bond is formed between amino acids 3 and 4. This peptide bond has extended the peptide chain by an additional amino acid. We've just added a new amino acid onto the chain, the amino acid that this codon called for, by the way. But now we're breaking some rules. This is now a peptidyl tRNA because it's holding onto a peptide chain. But it's in the A site, and peptidyl tRNAs are not allowed in the A site. They can only exist in the P site. So this is going to force translocation. The ribosome is going to move forward by one codon. It's going to shift so that the tRNA here, the peptidyl tRNA, is now in the P site, and the A site will be open. When translocation ends, the whole ribosome has moved forward by one codon. The next codon is now positioned in the A site, but everything is good again. We've got the peptidyl tRNA is in the P site, great. We've got the empty tRNA in the E site, fine. And then the process is going to repeat. The next new aminoacyl tRNA is going to come into the A site where it belongs, because it's only carrying a single amino acid. But since only two tRNAs can exist in the ribosome at any one time, this is going to evict the empty tRNA from the E site, and we're right back to the beginning. What's going to happen next is a peptide bond is going to be formed between these two amino acids. That's going to break this bond between amino acid number 4 and its tRNA. We're going to have a chain of 5 amino acids now linked to the tRNA in the A site, and that's going to break the A site rule. It's going to drive another round of translocation, which is going to put the empty 4 tRNA in the E site, and it's going to vacate the A site, and the process repeats. So here we can watch this happen in real time. We see a lot going on here. First, let's pay attention to the base pairing between the anticodon and the codon. We can see that each tRNA that comes in is base pairing through complementarity. The tRNAs coming into the A site are carrying single amino acids, the amino acids that are the ones encoded by those codons. Those single amino acids on those single tRNAs are aminoacyl tRNAs. They fit comfortably in the A site. But as the peptide bond is formed between the peptide chain and the P site, and the amino acid in the A site, we see that translocation is driven forward. Now we have to get the ribosome translocated so that the new peptidyl tRNA goes into the P site where it belongs. The empty separated tRNA moves into the E site, and it's bounced as soon as the next aminoacyl tRNA comes in, because we can only have 2 tRNAs in the ribosome at any one time. So that's the elongation of translation, the meatiest part of translation. Now let's kind of back up a little bit and talk about how translation begins and how it ends. So during translation, every incoming tRNA enters the ribosome through the A site, aminoacyl, right? Aminoacyl site for the aminoacyl tRNA. But obviously for translation initiation, that can't be. To start this process, we need a tRNA carrying a single amino acid to begin in the P site, and that requires initiation. So a specific tRNA called the initiator tRNA, this is the tRNA that can bind to the start codon. The start codon for every single protein on the planet Earth is AUG, and the anticodon that binds to that is CAU. So the initiator tRNA has a CAU anticodon, and it can base pair to the start codon. It's also the only tRNA that can enter the P site with only one amino acid on it, and it carries methionine. The only codon that methionine is encoded for is the start codon AUG. So AUG is the start codon. It marks the beginning of a protein coding region. The methionine tRNA recognizes that start codon. It binds to the small subunit of the ribosome, and the small subunit of the ribosome positions that methionine tRNA at the start codon and initiates translation. That small subunit keeps that messenger RNA, that initiator messenger RNA, engaged with the tRNA, and then we scan for that start codon. Once that start codon is reached, everything stably associates, and the large complex of the ribosome will now join the party. And it will join in such a way so that the initiator tRNA is in the P site. Once this has occurred, we're ready to go into the process of translation like we talked about before. The next tRNA will come into the A site. It'll be an aminoacyl tRNA. A peptide bond will form between the methionine and the amino acid carried by that tRNA. That will make that tRNA a peptidyl tRNA because it has more than one amino acid on it, which will drive translocation, yada, 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 yada. So again, the initiator tRNA is the only tRNA that can enter the ribosome at the P site directly with a single amino acid, and it's also the only RNA that can recognize a start codon to initiate translation. And then terminating translation is also a little bit straightforward. In both eukaryotes and prokaryotes, translation is terminated by stop codons, those 3 stop codons that I showed you before. Once we've reached the codon right before the start codon, up until that point, everything has happened appropriately. So the codon right before the start codon has a peptidyl tRNA on it. Peptidyl tRNA is in the P site. A site is vacant. But there is no tRNA that recognizes the stop codon. So the A site is vacant, but there's no tRNA that can enter because the codon in that A site, a stop codon, doesn't have a tRNA for it. Instead, proteins that are shaped to look like tRNAs, called release factors, enter the A site instead, and they bind to the stop codon. These are called release factors, and when release factors enter the A site and bind to the stop codon, they trick the ribosome into breaking this bond between the peptide chain and the tRNA in the P site. That frees the peptide chain, so it floats away, can now fold into its 3-dimensional shape and take on whatever job the cell needed from it. And that dissociates the ribosome, because the ribosome destabilizes once the peptide is released, and the ribosome is now free to recycle and go off and repeat translation at some other gene. So again, to summarize the process of translation, termination, we have a stop codon positioned in the A site. We're going to break the peptide chain away from the ribosome, away from the tRNA and the P site. That frees the peptide chain so it can go fold and do its job. This severing of the peptide away from the ribosome destabilizes the ribosome so that the small subunit, the large subunit, the tRNAs, and the release factor all dissociate, and they are free to recycle and do their job at another gene. I've posted a link to a short 2.5-minute YouTube video that goes into the details of translation quite accurately on Moodle, so if you want a refresher of this with all of the lengthy explanation, I encourage you to watch that video a number of times and look for all the features that we've discussed here. So that's it. As I warned you, a long video. Hopefully you did take some breaks in the middle of it, but this tells you the entirety of the molecular biology story, really in one nutshell. We talk about DNA as a physical molecule. Chromosomes are long packaged DNA molecules, packaged such that they can fit into a cell's nucleus. We discuss that packaging itself. Nucleosomes are the individual fundamental building blocks of packaged DNA. They consist of the histone octamer core, 2 copies of H2A, H2B, H3, and H4. And those octamer cores serve as spools. The DNA wraps around the outside of that spool to form a nucleosome. This bead on a string nucleosomal DNA structure can be further compacted by coiling it around a central axis, creating a 30 nanometer fiber, this coiled helix of nucleosomal DNA. And then we can further compact that 30 nanometer fiber all the way to mitotic chromosomes at 10,000 level compaction. We then moved from DNA as a physical molecule to DNA as a molecule of information coding. The first process of releasing the information from DNA is transcription, the process of copying a DNA gene into an mRNA form. Promoter marks the start site for transcription. In eukaryotic cells, we need general transcription factors to recruit and position RNA polymerase II at that promoter, and that requires the DAB-phase GTFs, TF2D, which contains TATA binding protein. It binds physically to the promoter. TF2A, TF2B, TF2H, which escorts in the RNA polymerase itself, TF2E and TF2H. TF2H phosphorylates the carboxy terminal domain of RNA polymerase II and releases it from the promoter so that transcription can occur. Again, part of TF2D is TATA binding protein, which binds to the promoter, to the TATA box of eukaryotic genes. Once transcription has been completed, the messenger RNA is not yet ready for translation. It first needs to be processed. It needs to get a 5' methylated guanine cap, a 3' polyadenine tail, and the introns must be spliced out by a spliceosome so that the exons are joined together. Now, that fully mature and processed messenger RNA can leave the nucleus and be translated in the cytoplasm. We read the code of messenger RNAs in triplets or codons, groups of 3 nucleotides that in that group of 3 encode the information or represent a single amino acid. The genetic code is our nucleic acid to amino acid dictionary. There are 64 possible triplets, 20 amino acids, and that allows us to have redundancy in the genetic code, which gives us a buffer against mutations so that sometimes we can have a mutation at the DNA level that doesn't change the amino acid the codon calls for and thus doesn't change the protein. tRNAs are our transfer RNAs, but they're really doing the job of translation. On one end, they have an anticodon, which can base pair directly to the codon in the messenger RNA. And on the other end, they carry the correct corresponding amino acid that that codon calls for. So tRNAs bridge codons in the messenger RNA to amino acids. Ribosomes are the coordinators of protein synthesis. They contain 3 sites, the A site, the P site, and the E site. And we've talked about the process of which tRNAs enter those sites and what drives translocation and the formation of peptide bonds. And we finally ended the lecture by talking about how translation initiates with an initiator tRNA carrying methionine that recognizes a start codon and enters the P site of the ribosome, as well as how translation ends or terminates when release factors enter the A site and bind to stop codons, disrupting the ribosome. And that is gene expression in 1 hour and 50 minutes. So, you know what to do. Let me know what topic or topics remain confusing in the dialog box that's about to pop up, and I will see you all on Friday to talk more deeply about this wonderful, wonderful topic of gene expression. After reviewing those complex topics, we'll hit this hard with some challenge questions, and then talk about an absolutely wonderful paper that goes into these concepts. So until then, thanks for watching, and have a great day.